So in this lecture, we're going to be looking at the mechanisms of hemostasis, the ways that the body will prevent the loss of blood. So hemostasis is the process that halts bleeding and stops up, closes off, seals up a damaged blood vessel. And there are three overlapping kind of phases or stages or steps in this process. The first is vasospasm, the second is platelet plug formation, and then the third step is blood coagulation. Vasospasm occurs as a damaged blood vessel, the smooth muscle layer of the blood vessel wall contracts, it spasms, and this is a reflexive response. It's initiated by pain receptors. So pain receptors notice that damage has occurred, and there's a reflexive response that immediately causes smooth muscle in the blood vessel wall to contract, and this will constrict the blood vessel, and that will either slow the loss of blood or may, if it's a small enough blood vessel or a small enough cut, actually stop the flow of blood altogether. And so here is an image of a blood vessel in the brain that here it is relaxed. So what we're looking at is this area right here, and this is what it looks like if that if that blood vessel spasms. And so you can see how much smaller the blood vessel becomes as a result of vasospasm. So if that doesn't stop the flow of blood, then the second step is platelet plug formation. So within about 15 seconds of an injury occurring, the platelets, the plasma membrane that surrounds the platelets will become very sticky. And so those platelets begin to adhere to any exposed fibers, especially collagenous fibers, protein fibers, that have been made available to, that the platelets have been exposed to following the injury. And so they respond to any rough surface, but especially collagenous fibers, by becoming sticky. And so when the platelet membrane contacts the collagen, then these spiny processes extend from the platelet membrane and adhere to the collagenous fiber, and the platelet actually changes shape. It becomes, it assumes a shape that will allow other platelets to attach to it. And so the platelets will accumulate there at the exposed edges of the vessel that's been damaged, and then they will adhere to other platelets, eventually forming a goodly sized plug. And if it's a large enough plug, like we see here in this drawing, that could potentially completely seal off the blood vessel. Here we see a clump of platelets beginning to form. And I'm not sure what they're adhering to, but you can get a sense of how they kind of adhere to each other from that image. Okay, so if vasospasm and platelet plug formation do not then seal off the damaged blood vessel, then we move into the third stage, which is called blood coagulation. And so about 30 seconds following an injury, the dissolved plasma protein fibrinogen will begin to be converted into a fibrous form of insoluble protein called fibrin. So fibrinogen is the dissolved plasma protein. Fibrin is the insoluble form, the non-dissolved form that is fibrous. And this will produce a web of protein fiber that will serve to capture red blood cells and platelets, and that web then will eventually clog, and that will prevent, and form a solid mass of protein fibers that are all wrapped up and clogged up with red blood cells and platelets. So coagulation, this process, is controlled by the balance of chemicals that cause coagulation, the procoagulant properties of the blood, balanced with the anticoagulant properties of the blood. And so if there's an increase in procoagulants that overwhelm those anticoagulant properties, then the blood can coagulate. And there are three primary events that occur during coagulation. The first is that a chemical called prothrombin activator gets made. So we have a series of chemical reactions, and along the way, prothrombin activator is formed. That prothrombin activator acts as an enzyme that will convert a material called prothrombin into the enzyme thrombin. And then finally, the last step is that thrombin will act as an enzyme that will cause a reaction to occur that converts fibrinogen then into the threads of fibrin. So thrombin is the last enzyme in the process. Prothrombin activator must be formed in order to get thrombin made from prothrombin. So as this tangle of fibrin fibers grows and gets larger, then platelets and blood vessels get trapped in that mass. And this produces a blood clot that will seal off the vessel that's been damaged. And all of this requires clotting factors. And that includes calcium ions. So calcium is important to this. And, got ahead of myself. And 11 different dissolved plasma proteins, as well as platelet factors that are released by the platelets that get that are in the area of the developing clot. And so it's this series of chemical reactions that converts thrombin, that first creates from prothrombin the enzyme thrombin, and then converts fibrinogen into fibrin. And so here we have, the image from the book that shows this series of chemical reactions. And there are two entry points. Well, let's look down here. So this region here is the common pathway. So regardless of how the process starts, regardless of how that process starts, we have to get into this common pathway to get coagulation to happen. And the common pathway begins with this material called factor X. And all these factors, it's very frustrating. They are not numbered in any reasonable order. They're numbered based on the order in which they were discovered. And so factor X was the 10th of these factors that was discovered. So if we can get a protein called factor X made, then factor X works to activate, along with calcium, factor 5 into prothrombin activator. So together, factor X, factor 5 working together, they produce prothrombin activator. Prothrombin activator will convert prothrombin into thrombin, and then thrombin will convert fibrinogen into fibrin. And factor 13 comes along here, and it stabilizes the fibrin, and eventually a clot forms. Okay, so we got to get, and by the way, I don't expect you to memorize these pathways. That's not what's important here. But what you should understand is that each one of these enzymes is reusable. So let's say we have factor X, and it's responsible for changing factor 5 into prothrombin activator. Well, once factor X, which is factor 10, I shouldn't call it factor X, it's factor 10. Once factor 10 does that work with one molecule of factor 5, it can do it again with the second molecule of factor 5. And so factor X, or 10 here, can produce lots of factor 5. Or, I'm sorry, I'm still messing this up, I apologize. Factor 10 can convert a lot of factor V into prothrombin activator. So we get a whole bunch of prothrombin activator made. Prothrombin activator can work its magic on a bunch of prothrombin. And so, if we get, let's say we get, 20x amount of prothrombin activator formed, well, we can get 80x, give or take, of prothrombin being converted into thrombin. So we can get a whole lot of thrombin from a moderate amount of prothrombin activator. So this is what then leads to this huge coagulation event, is because a whole lot of thrombin can be produced from a smallish amount of factor X. And this is just the ending point. So this common pathway is the end point. There are two mechanisms for getting into this common pathway, for producing lots of factor X. There is the extrinsic clotting mechanism and the intrinsic clotting mechanism. Extrinsic clotting mechanism occurs when damaged tissues release factor III. It's called tissue thromboplastin. Factor III works as an enzyme that will take factor VII and convert it into factor X if calcium is around. So that's one way. The second way is if a cut happens and the blood contacts a foreign surface like a collagenous fiber, or if there's a plaque in the blood vessel. We talk about that this is the artery closures as arteries get narrowed by plaque formations. So if the blood makes contact with a surface that it's not supposed to contact, then that will activate this enzyme called Hageman factor 12, which activates this factor 11, which activates factor 9. Okay, so a little bit of Hageman factor 12, because it can be reused, will activate a lot of factor 11, which will then activate even more factor 9, which in the presence of calcium will activate even more factor 8, which will then be converted into factor 10. So both factor 7 and factor 8 with different enzymes can be converted into factor 10. And then factor 10 now enters the common pathway, and we see this continued amplification of each step along the way. So again, I'm not looking for you to memorize this chart, but I just want you to get the idea that there are a couple of ways that coagulation can be achieved, or a couple of beginning points. Both of the beginning points wind up here at factor 10, which then is the beginning point for the common pathway. And that because there are multiple steps along the way, and that each time the enzyme can activate more and more of whatever the next factor is in the step, that we get this amplification. So again, if we have X amount of Hageman factor, we can amplify that, let's say, by 4 times, which in the next step gets amplified then when we compare it back to the amount of X that we had at the beginning. Maybe it's 16X, which then results in 16 times 16, 256. Well, maybe it's just 16 times 4. Again, I'm not trying to make this exact, but maybe we have 64X of factor 10 being formed, which then leads to 256 times the amount of X of prothrombin activator, and so on. So that when we produce thrombin, so much of it is made so quickly that it's like this huge, overwhelming chemical reaction that converts the plasma protein fibrinogen into fibrin, which then is stabilized and produces this clot. Okay. So there is my big, messy explanation. And what I'm really trying to get you to see is that it is when we get to the final step, it is overwhelming. And the blood will just, in a flash, convert from a liquid form to a solid, clotted up form. Okay, so once we have all of this damage kind of at least stopped, what follows next? So once this fibrin mass has appeared and we've trapped all those platelets and the red blood cells, well, the platelets then will contract. They become smaller. And this helps to pull together, to bind together the edges of the wound, and as that entire clot retracts, becomes smaller, and pulls closed the edges of the wound. The platelets release yet another hormone, a platelet-derived growth factor, that stimulates the smooth muscle cells and the fibroblasts that are there at the wound site to grow and to divide, grow and divide. And that results in repair of the damaged blood vessels. Fibroblasts move in from the surrounding connective tissue. They're going to produce collagenous and elastic fibers that will help to repair the damaged blood vessel. And as this repair is occurring, then the fibrin that is in that clotted area, as the blood vessel begins to repair, and as the tissues, the epithelial, the dermal, the subdermal tissues all get repaired, then the fibrin clot gets broken down by an enzyme that's formed from a plasma protein called plasminogen. The enzyme is called plasmin. So notice fibrinogen produces or undergoes a reaction where it converts into fibrin. Plasminogen is converted into an enzyme called plasmin that will break down the fibrin in the clot, and that way the white blood cells can move in and remove all of that clotted material. Okay, so what are some clotting disorders or some issues related to clotting? Well, okay, so here are some terms for you. A thrombus is a blood clot that can form on the inside of a blood vessel. And it can grow, it might block the blood vessel. And so we name these thrombus based on its location. So coronary thrombosis is forming in the blood vessels of the heart. A cerebral thrombosis will form in the blood vessels of the brain. A pulmonary thrombosis will form in the blood vessels of the lungs, and so on. An embolus is a circulating blood clot. So perhaps a thrombus will break free, or a portion of it will break free, and now we have an embolus. There are other things that can act as an embolus. An air bubble that's circulating around can act as an embolus. A fat glob that forms in the blood vessels could act as an embolus. And we name a chunk of plaque, that stuff that builds up on the interior of a blood vessel wall, that might break off and form an embolus. And we call it an embolism, if that embolus becomes lodged, clogging off a blood vessel. And it's, again, named by the location of occurrence. A cerebral embolism is a stroke. A pulmonary embolism can happen. A coronary embolism is a heart attack, and so on. Atherosclerosis is the accumulation of fatty plaques in the layers of a blood vessel wall or on the interior of a blood vessel wall. And that can cause changes in the lining of a blood vessel that may promote clotting. And hemophilia is a genetic disorder. It results in the production of non-functional blood clotting proteins. So it generally has what's referred to as a genetic etiology. So etiology is the cause. And so when I'm talking about blood clotting proteins, I'm talking about all those factors that we just discussed in the previous slide. So the severity depends on the specific gene or proteins that are involved. So remember, the entry point into the common pathway was factor 10. When it's produced, then that continues to have effects downstream in that flowchart that we were just looking at. But because factor 10 is a common pathway protein, then that's going to cause really severe hemophilia. Whereas if one of the the factors further up in the flowchart is what is affected by this genetic disorder, then there may be less effect to the person. Okay, then last of all, we've seen how clotting could happen, but clotting, if it happens inappropriately, can form an embolus and then an embolism. So how do we prevent that kind of clotting from happening? Well, the lining of the blood vessel, that endothelium, is very slick and smooth. And so that slickness makes it difficult for clotting to begin inappropriately. The cells there of the lining of the blood vessel produce a hormone called a type substance, a hormone-ish substance called a prostaglandin that will help to prevent the platelets from adhering to the blood vessel lining. As a blood clot forms and those fibrin threads develop, the protein threads themselves bind up the enzyme thrombin, and that helps to prevent the enzyme from spreading beyond these damaged tissues. So it's not just circulating free in the blood and spreading far away from the site of the developing clot. Instead, it gets trapped there. I'm sorry, I got the yawns all of a sudden. And is held there in the damaged tissue. There is another plasma protein called antithrombin that will inactivate thrombin so that it won't get out of control. And then finally, mast cells and basophils. Mast cells are found in connective tissues. Basophils are a kind of white blood cell. They secrete heparin, which blocks the production of prothrombin activator. It can interfere with the activity of thrombin, and finally, it will inactivate, heparin does, it will inactivate and remove thrombin from the blood. Okay, so there you have everything I've got to tell you about coagulation. Any questions? Send me an email. See me in office hours.